This is a production of Cornell University.
Yeah, thanks, Adam, for the nice introduction and also great invitation. It is really a great pleasure to visit Cornell and see many good friends and see Greg for many years. We met when I was a first-year graduate student back in Kansas. So it's really impressive visit this morning to see many of you. So today I'm going to present some of work after I landed my job in Texas. So I titled my work on the non-self recognition and signaling mechanism in plant innate immunity. The fact is, although we seemingly live in a very peaceful, wonderful world, but in fact, we are surrounded by all kinds of enemies. It's a dangerous world, right? So bacteria, fungi, viruses, and insects. And the fact is, we're all healthy most of the time. And resistance is the rule. Disease is rather exception. This is in large attributed by the robust innate immune system. So at the heart of the immune system is the individual capacity to recognize self versus non-self to elicit the defense response. So the key to a healthy immune system is its ability to distinguish between the body's own cells and recognize them as self and foreign cells as non-self. So this echoes a famous saying I learned when I was an elementary school student from Shunzi, a famous Chinese philosopher. So knowing your enemy and yourself, you can fight without defeat. So compare with the animals, plants even more vulnerable. They are sessile when they get sick. No way to talk to doctors to get rescue, right? They also don't have the adaptive immune system as the animal system. And they also don't have the specialized somatic immune cells. And what they share is this robust innate immune system. So the innate immune system in plants has been classified as a two-tiered defense system. The perception of perception of microbial components by cell surface receptor trigger the first line of defense response, also termed as PRR, pattern triggered immunity. For short, it's PTI. So those microbial components collectively, they were termed as PAMP or MEMS, stands for pathogen or microbe-associated molecular patterns. And pathogen infection also often trigger damage to the host cell, which generate endogenous damage-associated molecular patterns. So collectively, those things can be perceived by the cell surface pattern recognition receptors. So in plants, what is striking is we know those microbial components are perceived by receptor-like protein or receptor-like kinase. And in plants, there are up to several hundreds of these kind of proteins. In contrast, animals, for instance, human, up to 10 TLR proteins perceiving those microbial components. So this may provide sorts of for plant cell surveillance system to recognize all different kind of microbial components to provide this fundamental basal defense response. So upon perception of microbial components, plants trigger complicated signaling pathways. So those signaling cascade has been classified as two-tiered. One is ETI and the other one is PTI. PTI is mainly mediated by cell surface receptor, while ETI is mainly through the intracellular receptors. So today I'm going to present some of our work in the context of known knowledge in PTI signaling and demonstrate up to now how we think those PAMs are perceived by the receptors, how the signaling was transduced up to the headquarter nucleus to activate immune gene or defense-related gene expression. So one of well-studied PAMP is from bacteria-derived fledgling. So bacteria use this motor engine flatula to swim around. It's an essential component for bacteria. Its major protein consists of fledgling, and some of part of fledgling protein can be recognized by the host. For instance, the 22 amino acid peptide corresponding to an N-terminal region of fledgling could be recognized by Arabidopsis receptor FS2 which encode a leucine-rich repeat receptor-like kinase. So upon the fledgling perception or FLAG22 perception, FS2 protein quickly, almost instantaneously associated with another RLK similar like FS2 called BAC1 form a complex. So recent structure study also demonstrate both FS2, BAC1 kind of mediating the recognition of bacterial fledgling trigger downstream signaling events. So upon the bacterial fledgling recognition, a series of immune response can be detected by various ways. For instance, the rapid activation of evolutionarily conserved MAP kinase signaling cascade by this MAP kinase antibody Western blotting, or defense-related immune gene activation by RT-PCR, or the redox changes, for instance, the reactive oxygen species production as early as a few minutes. And also later, you could observe chylose deposition, which further lignify the cell wall to protect the cell from further infection. And also prolonged treatment of fledgling or the FLAG22 peptide inhibit the Arabidopsis seedling growth, likely caused by constitutive activation of defense response. And also pre-treatment of this fledgling of FLAG22 peptide protect the plants from the further secondary infection of the burdens pathogen. So one perspective we examined in order to understand how fledgling trigger immunity is transduced is looking at the phosphoproteomic dynamics and transcriptome changes upon FLAG22 treatment. Through these two series effort, we found one small kinase originally called botrytis-induced kinase BIC1, which encode a receptor-like cytoplasmic kinase, could be quickly phosphorylated upon FLAG22 treatment. As shown in this Western blotting, the band kind of migration got delayed. Later, we did a series experiment to demonstrate this indeed is a phosphorylation event. For instance, treating the sample with phosphatase CIP could block this band shift. So indicating this protein is responding to FLAG22 treatment. And this small protein, although encode a cytosolic receptor-like kinase, but it has a putative merstolation motif. It's targeted to the plasma membrane. And the FLAG22 receptor FS2 and BAC1 also a plasma membrane localized protein. Then we further examined whether they tend to physically associate to each other. So this is a co-immunoprecipitation assay. You could see BIC1 form a complex with FS2 and BAC1 without the ligand perception, suggesting they form a pre-existing complex. And interestingly, upon FLAG22 treatment, seems like the BIC1 got released from the FS2 and BAC1 complex. And both BIC1, BAC1, and FS2 encodes kinases. So we further ask whether there are any phosphorylation events occurs within the complex. here shows you actually from in-gel kinase assay, you could see actually BAC1 could strongly phosphorylate BIC1. And we further mapped the major phosphorylation site. If you mutate this site, you largely compromise the phosphorylation events. And also, we demonstrate, in addition, BAC1 phosphorylate BIC1. BIC1 also transphosphorylated back to BAC1 to further enhance the kinase activity of BAC1. And so for plant, RLK family has been classified as serine kinases. So along with our identification of phosphorylation sites of BIC1, we found actually BIC1 has multiple sites. It's phosphorylated at the tyrosine site. And then we mutate some tyrosine site and find one of the tyrosine site is particularly important to mediating the kinase activity for both serine activity and the tyrosine and the total kinase activity. But some site is specifically required for tyrosine activity, not for serine kinase activity. And we further asked whether those tyrosine phosphorylation residue are required for BIC1 function in defense response. You could see mutation of those tyrosine sites no longer able to complement the BIC1 mutant phenotype as the wild type BIC1 in terms of botrytis infection and ROS production. So suggesting those tyrosine sites is important for BIC1 function. And along with the recent study for BAC1, actually from Stephen groups, they demonstrate BAC1 also potentially carry auto tyrosine phosphorylation activity. We further found actually BIC1 phosphorylation tyrosine site is mediated by BAC1 directly. So this research is suggesting RLK complex tyrosine phosphorylation maybe is a prevalent event rather than previously predicted, mainly mediated by serine kinases activity. So as I mentioned early, so upon FLAG22 perception, FS2 from a complex with BAC1. BAC1 originally was identified as a brasinolide hormone BRY1 receptor associated kinase. So BAC1 formed complex with BRY1 play important role in BR-mediated growth regulation. So our research demonstrates BIC1, the small kinase, function as a direct substrate of BAC1. Then recent study also demonstrate there is extensive crosstalk between BR signaling and FS2 signaling. Although the mechanism is still not clear, we know the BR-mediated signaling activation unidirectionally suppress the FS2 signaling at multiple steps. So then we asked what kind of role BIC1 play in BR-mediated signaling transduction. So to our surprise, so BIC1, in contrast as BAC1, play a negative role in BR signaling. As you can see, this is a typical BR response assay. Unlike BAC1, BIC1 is insensitive, even hypersensitive to BRG, which is a BR biosynthesis inhibitor. And similarly, in terms of the response to BAS1, which is a key transcription factor in BR signaling, once BR signaling is on, this transcription factor is dephosphorylated, as shown from the migration shift from the Western blot. And you can see in BIC1 mutant, both the basal level and upon BL treatment, the BAS1 dephosphorylated form increased. So this also, consistent with this BRG treatment result. BIC1 has BIC1 mutant has elevated BR signaling going on. And similarly, the BL suppressed gene is further suppressed in the BIC1 mutant. And we further crossed the BIC1 to the BRY1 mutant. You could see BIC1 elevated the growth defect of BRY1, the BR receptor phenotype. So genetically suggests indeed BIC1 play roles and function downstream of BRY1. And we further explored the potential mechanism why BIC1 plays such kind of role in BR signaling. And similar as FS2 and BRY1 also encode LR type of receptor-like kinase and sitting on the plasma membrane. Here, the co-IP assay, you could see BIC1 also associated with BRY1 from a pre-existing complex. Interestingly, upon BL treatment, BIC1 also is released from BRY1 complex. This is strikingly similar as the complex dynamics of BIC1 with FS2 in PTI signaling. Although BIC1 plays different role, in PTI is a positive regulator, but in BR pathway is a negative regulator. And this BIF assay further confirmed the association of BIC1 and BRY1. And we also know BRY1 is a strong kinase. We ask whether there are any transphosphorylation events between BRY1 and BIC1. As you can see from the in vitro kinase assay, actually BRY1 strongly phosphorylates BIC1, even stronger than phosphorylation towards BAC1. This is the initial study for BRY1 and BAC1 phosphorylation mediating the BR signaling. And then, interestingly, upon BL treatment, it seems like a BRY1 phosphorylation towards BIC1 is further enhanced. So suggesting upon BL treatment, there is an enhancement of BRY1 phosphorylation towards BIC1. Then we further asked the consequence of the phosphorylation between these two components. As you can see here, neither the BIC1 kinase mutant or BRY1 kinase mutant, which both of them lost kinase activity. You could see they still form complex. The BIC1 kinase mutant still form complex with BRY1, and the same thing for BRY1 kinase mutant with BIC1. However, they no longer released from the complex. So seems like the complex become very tight if there is no phosphorylation events, suggesting the phosphorylation between BRY1 and BIC1 maybe is required for this complex, required for dissociation of BIC1 from the BRY1 complex. So through this research, we demonstrate that this BIC1 play negative role in BR signaling through directly phosphorylation of BRY1 towards BIC1, and somehow negatively regulate BR-mediated development and growth. However, BIC1 directly phosphorylated by BAC1, positively regulating the PTI response. So we still don't know exactly how this distinct, very likely is distinct phosphorylation coming from BAC1, from BRY1, dictating such kind of different outputs. We are mapping the differential phosphorylation for BIC1 through upstream of kinase to further address this phenomenon. And another perspective we explore FS2 signaling is based on the observation. Actually, BAC1, as I said, form a complex with FS2, also form a complex with BRY1. Recent several years, we found actually BAC1 also is required for additional other PAMP-mediated responses, including BAC1 form direct complex with EFR, which recognize bacterial EFTU PAMP, and also form a complex with DAM receptor type R1 and 2. So we are thinking, so any component associated with BAC1 potentially can serve as a conversion point to further mediating the PTI response. So we look for the BAC1 associated protein. Through this line of research, one particular protein we focused is a plant U-box containing E3 ligase called PARP, a particular PARP 12 and 13. You can see in the E2 hybrid, BAC1 and PARP13 directly interact, as so in the co-IP assay. And unlike so the BAC1 and PARP13 interaction is constitutive without ligand already form a complex. However, it seems like the PARP13 associated with FS2 only upon the ligand perception. And this complex formation occurs extremely fast, as early as a couple of seconds. We could see by co-IP experiment FS2 PARP13 form a complex. So this data suggests maybe this PARP E3 ligase associated with BAC1 upon ligand perception of FS2, so somehow they quickly form a complex. Then the PARP encodes a E3 ligase. We ask whether this E3 ligase does anything to do with the component in the complex. We performed the in vitro ubiquitination assay. To our surprise, the PARP13 directly ubiquitinated the FLAG22 flagelline receptor FS2, as you can see from this gel, the LADA formation. However, it did not ubiquitinate the co-receptor BAC1 or BIC1. So this may also suggest the signaling specificity, as this one is the primary receptor for flagelline. The other kind of play more pleiotropic role in multiple signaling events. And we also know ubiquitination of protein can mediate either for protein degradation or send the protein for cycling to activate the signaling. Then we further ask, what's the consequence of this E3 ligase mediated ubiquitination? Here you can see with prolonged FLAG22 treatment, the receptor FS2 underwent the degradation. However, in the PARP mutant, particularly in the double mutant, such kind of degradation is largely reduced. So suggesting genetically, providing genetic evidence, PARP12-13 mediated ubiquitination on FS2 likely is mediating FS2 degradation. And genetic analysis of PARP mutant further suggests support our hypothesis. You can see in the PARP mutant, the FLAG22 induced callus deposition is further elevated compared with wild type. Similar for disease resistance, the PARP mutant is more resistance compared with wild type, so suggesting PARP play a negative role in PTI response. Then this PARP protein encodes a modular protein, so has an N-terminal uncategorized but a plant-specific domain. And then in the middle is the U-box domain, and then C-terminal has an armadillo repeats. And we further did a series deletion mutational analysis to narrow down actually this armadillo repeat is mediating the specific interaction, directly interaction with BAC1, and also phosphorylation mainly occurs on the armadillo repeat by BAC1. And then the ubiquitination towards the substrate FS2 mainly mediated by the U-box domain. Then we hypothesize whether we, by generating the armadillo repeat domain alone, will generate a dominant negative version of this E3 ligase, will block the access of the substrate, could generate the dominant negative version of the PARPs. So we generate the transition of all expression, the ARM domain alone, transition plans. you could see these transgenic plants behave similar as the PARP mutant. Particular, it reaches to the similar level as the double mutant. So the transgenic plants become more resistant to bacterial pathogen infection and ROS production. And also you could see from the MAP kinase activation, the PARP single mutant or the double mutant show more MAP kinase activation compared with wild type. Basically, the transgenic plants is similar as the PARP double mutant. So suggesting this PARP O expression arm domain alone could provide a dominant negative effect. The reason we do this is it's a challenge to study the individual function of the PARP because of the redundancy. This is a large gene family. So by doing such kind of dominant negative thing, we wish we could further elucidate additional members of PARP gene family's function in immunity and other process. So with this line of research, it seems like once the defense response is activated, the cell has a mechanism by recruiting the E3 ligase into the receptor complex, and then quickly degrade the receptor to downregulate the immune response when it is no longer needed, right? So to keep the defense response tightly under control. So this again echoes a famous saying I learned from elementary school from the same person. So there is no instance for any country benefiting from a constant defense process, right? So at the right time, we should stop such kind of things to benefit the society in the long run. So stop the war, we should get support from government for research. That's, yeah. So meanwhile, I also view this line of research, although we and the plants look definitely, different. My daughter at elementary school knows, oh, you are a fake doctor. Although you work at a Mass General Hospital, you are not a true doctor. You work with plants. It's different. But I'm trying to tell her what I do is really important for real doctors. Because you see what I found is marvelously similar as what happened to us. We, as human and plants, we all use a similar type of protein. Those receptor proteins sit on the plasma membrane. Arabidopsis use this FS2, and human use TLR4 for LPS, TLR5 for fledgling. And then the downstream for activation of the signaling cascade. For human, we have the Iraq site of kinase. And then the similar type of kinase, big one, BAC1, transduce the FS2 signaling. And then we further found that the E3 ligase monitor the defense latitude by E3 ligase working on this receptor part. And similarly, in human, we have TRAS3A, exactly same looking E3 ligase work on this TRAS4 to monitor the defense response. And in addition, in human, there are another type of E3 ligase, and we monitor this Iraq kinase to fine-tune the immune response. Recently, we found that actually there are similar type of E3 ligase also working at this level. So I tell her how important we are. So, so this is, we are taking the proteomic approach to look at a genome-wide level, how the ubiquitination potentially regulating the PTI response. So what we did is to purify the ubiquitination associated protein, and then do mass spec and to identify such kind of protein. To our surprise, out of our list, from the receptor to RCK to the MAP kinase level to the transcription factor level, those known components are on our list. So at multiple levels, defense response are tightly checked. by such kind of mechanism shared in human and in plants. We are in the process, particularly interesting for this protein, you see the ubiquitination. This gel, as long as you can see signal indicating ubiquitination events, so you could see this protein is specifically induced upon FLAG22 treatment. So we are in the process to further characterizing what's the E3 ligase involved, what's the consequence to monitor this protein for ubiquitination process. And another approach we took to understand the PTI signaling is the classical genetic approach to looking for additional components involving PTI. So the philosophy is we use the early PTI marker gene as a readout. So here we choose FRK1, so-called, which encode a flagellating-induced receptor kinase 1. So this gene can be induced by FLAG22 very rapidly within 30 minutes. So we clone the promoter, hook up with luciferase, and generate the transgenic plants. So basically, these transgenic plants, if you treat the plants with flagellin or non-pathogenic or non-host bacteria, these plants light up. And then we mutagenize these plants and then looking for mutants, either hypersensitive or no response. And looking for mutants, we name those mutants as ardosis gene governing immune gene expression as Aggie. You know, we are, our logo is Aggie, so this I attract a lot of undergraduate students and a kind of free labor to do the initial screen. But turns out it's a fun project, and so the first 6 mutants right now we are characterizing and are coming from one RU undergraduate student. It's really fruitful screen. So one mutant I share with you, sort of we kind of understand how this mutation causes such kind of phenotype is Aggie1. You could see Aggie1, actually we have 2 alleles later. We map-based cloned both of them and found they are mutated as the same gene. And so here I showed both of you, both them together. You could see the Aggie1-1 or 1-2, the luciferase are much higher than wild type. And also this mutant in response not only to FLAG22, but also other PAMs, for instance, here ERF18 are non-pathogenic or non-host pathogen, suggesting the mutation not only, very likely is not affecting the receptors. It's something happening more downstream because we know different PAMs likely perceived by different receptors. We also performed the RNA-seq analysis, and seems like the Aggie1 mutant has further enhanced FLAG22-induced gene transcriptome changes. And this mutant also show enhanced disease resistance to virulence bacteria. And also, strikingly, this mutant is extremely resistant to polymildew or fungi, or fungal pathogen. However, this mutant show unaltered FLAG22-mediated MAP kinase activation. The ROS production also seems like similar as wild type. So suggesting the mutation occurs either downstream or independent of MAP kinase activation or ROS burst process. So we further perform map-based cloning and clone this gene. Actually, Aggie1 encodes CPL3, which is a putative CTD phosphatase-like protein 3. So what is CTD? CTD is the RNA polymerase 2 C carboxy terminal domain. It's a RNA polymerase 2 C terminal domain. So we know RNA polymerase 2 is an essential component regulating all the gene transcription process, right? Then this RNA polymerase 2, the C terminal domain consists of various repeats of conjugate peptide. So in different organisms, these repeats number varies. In Arabidopsis, there are 32 or 34, depends on how you classify these repeats in terms of conservation. And in human, seems like with more complicated organism, the repeats number increase. But each individual repeats consists this very conserved 7 amino acid peptide. And among these 7, 5 of them could be phosphorylated, consisting serine and tyrosine and threonine residue. So through many elegant research in yeast, we know the wave of phosphorylation, the dynamic phosphorylation process on this individual residue, on these multiple repeats, orchestrate the transcription process through the initiation, elongation, and termination process. So our mutant encodes a phosphatase. In yeast, the corresponding homologue in yeast has been shown to dephosphorylate certain residues of these heptap repeats. So then we've we examined whether there are any response for RNA PAL2 upon FLAG22 treatment. So we cloned the C-terminal domain of PAL2 and put it in the cell. You could see strikingly upon FLAG22 treatment as early as 2 minutes. And this CTD, RNA PAL2 C-terminal domain, show rapid mobility shift. Later, we did more work demonstrate this mobility shift is a phosphorylation event. And here, this gel is using the phosphor-2, SIR7 antibody to detect the endogenous RNA PAL2 phosphorylation status. You could see upon FLAG22 treatment, this various residue responding to FLAG22 treatment got active phosphorylated. Then we further asked what kind of kinase acting upstream to activate the phosphorylation of PAL2 CTD domain. So in yeast, we know the cyclin-dependent protein kinase can mediate the phosphorylation activation of the CTD. In particular for the CDKC8 family. And then we're looking for the corresponding homologue in Arabidopsis, and so which is the closest member is CDKC1 and 2. And again, to our surprise, particularly for CDKC1, upon FLAG22 treatment itself is soul migration shift again. Later we did more work and demonstrated this again is a phosphorylation event. So indicating the CDKC1 is responding to FLAG22. It's activated upon FLAG22 treatment. And we further asked what's the upstream of kinase regulating the CDKC activation. We know in the PTI cycling, right below the PRR complex is the convergent MAP-kinase cascade. We explore whether MAP-kinase play important role in activating of the CDKC cascade. And we use the MAP-kinase specific inhibitor or the MAP-kinase phosphatase. You could see this inhibitor or the phosphatase largely reduce the activation of the CDKC. So suggesting maybe a MAP-kinase cascade working upstream of CDKC activation. And we know that in Arabidopsis PTI cycling, MAP-kinase 3, 6, 4, and 11 play important role in PTI pathway. And we further test which specific MAP-kinase activate the CDKC. Here, from this immunocomplex kinase assay, you could see MAP-kinase 3 could directly activate CDKC. And meanwhile, we also see MAP-kinase 3 also activate CDKC2. And we further performed mass spec analysis, identify the phosphorylation site of CDKC, and you could see mutate this phosphorylation site, you wipe out the MAP kinase activation towards CDKC. And as I said, the mutant we isolate encodes a phosphatase, and it's a putative phosphatase. So only in yeast it has been shown this is a functional phosphatase. Then we further demonstrate actually the one in the adopted genome also indeed carry a phosphatase activity. And here shows you when you have the catalytic motif of the phosphatase, you could wipe out the phosphorylation of CTD activated by CDKC upstream kinase. So taking this together, we demonstrate through the genetic approach, we find actually downstream MAP kinase, there is a CDKC complex could directly phosphorylated by the MAP kinase cascade. And then this CDKC directly phosphorylate the RNA polymerase II C-terminal domain to activate the PAL2 transcription orchestrating the immune-related gene expression. Well, this phosphatase we isolated from the genetic screen could dephosphorylate the PAL2 C-terminal domain to counter-regulate the PAL2 activation on the immune gene activation. I spent a few minutes to explain a little bit for my domestic project on cotton after I moved on to Texas, which is the largest acre growth for cotton. So cotton, when I do risk, initially I check cotton as a crop is really cool crop, all smiling face with cotton fiber. Everybody loves cotton. Well, if you use cotton research, do a similar type of word search, you come up with these two things everybody said, because it's a very difficult and complex system, and we see extremely long growth cycle and difficult for transformation. Only a few cultivars it's able to be transformed. So, but meanwhile, it's important. So I thought we should do something with cotton, particularly with local potential funding support, etc. So the way we tackle cotton is to develop a transient assays, because so far for cotton, most of research focusing on genome evolution perspective of fiber development process. There is any known origin has been isolated from cotton. And my base cloning is a pain of neck. The genome is 2.5 kb. It's allotetraploidy. It's very difficult. So the transgene assay we developed, one is loss of function by aggregated Weeks assay. Another one is gain of function assay through ProPlus-based transgene transfection. So I can escape the mechanism for Weeks. But importantly, we found the TRV-based virus vector works perfectly in cotton. You could do the aggregated infiltration 10-day-old seedling and observe very efficient silencing at 2 weeks post-inoculation. So importantly, this Weeks works on all different kinds of cotton cultivars. So far we tested a commercial one, and everything grow in the land. So this really gave us the power to potentially explore the origin things. So what to explore the novel gene in cotton, what we did is we built the VGS library and then try to perform similar kind of screen like a lot of people did in tomato and tobacco. So inoculate the individual agrobacteria with the individual seedling and then screen for different phenotypes. And then we can go back to sequence the agro we used to resolve which gene causes such kind of phenotype. If this works, this is going to really revolutionize the cotton MAP-based cloning process. It's a long process, and so far there is no any succeed in cotton yet. And so as a molecular biologist also, we are not satisfied with only see the phenotype. We want to understand why this happened, right? So it is very important to have a system allow you to put the gene back to the system to look at what happened. So I modified the procedure for ProPlus system. So this is really allow you to put the gene back in 2 days, and then you can do all sorts of molecular biology to look at what is the protein behavior and to study the biochemistry part of the gene function. And center on these two platforms we developed, we focusing on verticilliar wilt, which is one of devastating disease in the field worldwide. And particularly with the VE1 and BAC1, they all PRR type of like FS2 type of receptor like kinase, examining the signaling transduction in this cascade. And more recently, we are also doing collaboration work with A&M for bacterial blight, looking for tail targets mediating either resistance or susceptibility. And another perspective is root rot. Root rot is very bad in Texas. And so we're developing the VIGS in the root part, so allow us to silence the gene in the root, examine the requirement mediating root rot. Another thing is the drought tolerance. The reason I did the drought is, initially drought is one of the easiest trick to do. We just do inoculation and stop water them, and then go back to root. So although, with kind of successful in terms of isolating two components, although they are in the non-ABA pathway, but there is no molecular link. Seems like we are building something quite interesting. One of MAP kinase serving as direct substrate of SNRK kinase. And SNRK kinase also kind of directly phosphorylate MAP kinase, which is something we don't know in the previous knowledge. And these two pathways work independently in the downstream of dehydration pathway. So we are trying to figure out the exact mechanism why we saw they are connected, genetically also independent. So share a little bit the preliminary result we did together with A&M. It's a really fun project. And so the tail effect, actually for cotton bacterial blight, it's a seedling disease. So through many years of research, we know the causal agent sensomonas, the main pathogenicity factor is mediated by type 3 factors. And among those type 3 factors, a larger group are tail effectors. And we know the tail effector function like a transcription factor. It carries the nuclear localization, activation domain, and also has a unique DNA binding specificity repeats. Those repeats dictating the recognition specificity of the host target sequence. So through Adam's research, we know that we can potentially predict the recognition side of the individual tail effectors. So then we choose two typical such kind of tail effector. One is AVRB6, which in cotton is one of the well characterized in terms of the corresponding resistance loci. And another one is PTHN. It's the major burdens determined in sensomonas. So we clone those individual tail effectors, express them in the cotton ProPlus cell, and then perform the transcriptome analysis of upregulated or downregulated genes by these two tails. And then working together with Adams to predict the potential target gene among those altered expression transcript. And then we further confirm those transcription changes and then perform a series experiment in terms of the transactivation assay, and also next step is to perform large-scale kind of VIGS assay or detailed assay to examine the requirement of those candidate gene mediating AVRB6 mediated HR resistance and PTHN mediated susceptibility. So hopefully through this, we can provide information what kind of host target mediated by these AVR or PTH effectors from sensomonas. So with that, I would like to thank folks that did work in the lab. and also wonderful collaborators, a lot of bioinformatics work together with Zhang Junfei here at BTI, and Adam, of course, and several other people in various institutions. Those are funding agencies. I'm sorry to give such long talks. I'm supposed to stop 5 minutes early. So now I'm happy to take any questions if you have.
This has been a production of Cornell University, on the web at cornell.edu.